We are talking through today the homework questions, and then we may be able to actually start in the next unit. We'll see how far we go. But at least at this point, we should be able to walk through and talk out answers to the chapter 5 and 6 questions. So that's really what I'd like you to start with today, is just pairing off or grouping off and talking those through. I'll assign questions momentarily. And that'll be our main goal for the day. Sound good? Any questions? All right, let's do that. And I will turn the lights down as we usually do also. There we go. So hopefully you can see. Okay, so the first one, I've always really loved this story. It's the classic, like, you know, scientist wakes up in the middle of the night, rushes to the lab to solve some problem that he's been working on for a long time. So I hope you enjoyed the box and the story about it. I think it's also just a fun little experiment to model. So let's just sort of work through it. So the first one, I guess all three of these went to Anna. So what did isolating the hearts and the vagus? So he isolated the hearts and vagus nerves of the two frogs, frog A and frog B. What did he do? Yeah, go ahead. Okay. Okay. That last one was kind of, whoops. I need to start a new document. That last one was kind of wide open. So did anybody have any other controls they thought of that would have been useful? So you've got two frogs sitting on the surgery table. They both have their hearts open to you, and you stimulate one of their vagus nerves, and then you move saline from one to the next. Maybe we'll come back to that, because I'm sort of leading straight to prediction here. So prediction, this one went to, who did this go to? Oh, all these went to Sydney. So what did you get for your prediction for what should happen if you move saline to frog B? Yeah, so like I'm assuming you capture these neurotransmitters, the chemicals, the soup, as they put it, as opposed to the sparks. Assuming you capture these neurotransmitters, then frog B's heart should slow down when you add the saline that has those neurotransmitters to them. Right? So that's what should happen, and what is in fact to happen. What were the actual results? So the acetylcholine was captured in that saline, which to me is borderline miraculous that there was enough acetylcholine that got in that saline, and it was at high enough concentration, they still was able to have some sort of an effect. But it did actually work. Frog B's heart slowed down, despite the fact that Frog B's own vagus nerve wasn't doing anything. It was all acetylcholine that came out of Frog A's heart, or Frog A's vagus nerve, that is causing the effect on Frog B. So hypothesis supported? Yeah. This is one of these experiments where I feel like if it hadn't worked, I don't know, like there's all kinds of reasons it might not have worked. I still just think it's amazing that there was enough acetylcholine that got in this saline that he was able to move it from one frog to the next, just by, basically rinsing the heart and then capturing that neurotransmitter. But it was enough, and it did actually work in this case. One other thing, I agree as far as this control. I think you definitely need to know that. I think there's one other potential problem that you could run into, and that could be that if you just squirt pure, I think you would want to do a trial where you have frog B sitting there and its heart is plucking along, and I think you'd want to do an experiment where you squirt some just raw saline without neurotransmitter on it, just to make sure that the act of squirting saline doesn't slow the heart down. Because that could happen. Like it could be nothing in the saline per se. It might be that there had nothing to do with acetylcholine being in the saline. It might just be something about bathing the heart in additional saline that would cause it to slow down. So I think I would do this one, but I would also say an additional control. Yeah, so I think it would also need some kind of an additional control in which you bathe the heart in saline without the acetylcholine neurotransmitter that we presumably captured by stimulating frog A. We'd want to not do that. We just want to bathe it in some saline, and that way we make sure it's not the act of bathing in saline or something else about the saline that's causing the heart to slow down. We want to make sure it's that neurotransmitter that we captured from frog A and moved to frog B that's really doing it. I've always wanted to do this experiment. And it's not that hard of an experiment to do. So I don't know, someday, maybe. I just don't want to do that to the frogs is my problem. So. All right, let's do number 2. So number 2 says explain the difference between spatial summation, temporal summation, making explicit reference to figure 5.19. This one went to Taylor. Are you there? Okay. Agree, disagrees? That all look good? These are, I think these are fairly easily confused because I think people get caught up in the term. But basically the idea is you've got, so spatial summation, you're summing across space. And you got two things happening exactly at the same time. Temporal summation, you're summing across time. So if you're recording, like if you had a single EPSP, that would just be, kind of a little bump like this in your recording. So this is membrane potential, this is time. If you had temporal summation, you'd expect to see sort of two right in quick succession. So you'd probably be able to detect them. They'd probably look like, and then maybe another one right on top of that. So that'd be temporal summation. So you got two EPSPs that are like stacking on top of each other. This one would have gone down like this, except that another one stacked on top of it. Spatial summation, you really couldn't see them because they happen at the same time. It would just be like, like that. Because you basically have one and then another one stacked on top of it. But the key thing about it is that this is, well, I'm about to spill the beans, so I'm not going to do that. So it says, the next bit says, use the discussion of quantal analysis to explain why spatial and temporal summation are really important for CNS neurons, not so much at the neuromuscular junction. So, I'm sorry, who had this one? Sarah, you had this one. What did you get? Yeah, did you get to think about quantal analysis? Like, did you see what that? Yeah, that's sort of an additional bit on top. But I mean, it's very much, I think, in line with what you have here. So the idea is, like you said, neuromuscular junctions, those synapses always work with just one stimulation. And the reason is they have a huge amount of neurotransmitter that's released. So they release a huge amount of neurotransmitter with each presynaptic action potential. Just massive, massive amounts. And so the result is that the postsynaptic cell gets a massive amount of depolarization from all that neurotransmitter coming across. And so CNS synapses, on the other hand, they often, if you get an action potential in the presynaptic cell, they will often only release like a single vesicle worth of neurotransmitter. Just pop, one little vesicle. So there's one little batch of neurotransmitter that goes across. Whereas, you know, it's like hundreds of vesicles being released from the neuromuscular junction with each action potential. Quantal analysis, the bit about quantal analysis is that it tries to, the idea with quantal analysis is that you can actually quantify what is the impact of one vesicle on the postsynaptic potential. Oftentimes there's sort of a rough estimate that one vesicle might result in 0.1 millivolts worth of a depolarization, something like that. So if you're only giving 0.1 millivolts of depolarization to the postsynaptic cell, that's usually not going to be enough to get you up to threshold if you're in the central nervous system. In the neuromuscular junction, you have, you know, each vesicle maybe is worth 0.1 millivolts, but you're releasing 100 of them. And so you're going to get up to threshold in the neuromuscular junction because there's so many of these vesicles being released with each action potential. But in the CNS, that won't happen. So the only way that in the CNS that you're ever going to get up to threshold, which is, you know, at least 5 millivolts or so above resting, is if you have a bunch of these vesicles being released at once. And the only way you get that to happen is if you have temporal summation, so the same synapse is releasing vesicle after vesicle with each round of action potential that hits. Or if you have spatial summation, where this is often the case, where you've got multiple input neurons that are all firing and targeting the same target neuron simultaneously, and they're all, so you get spatial summation happening as well. Does that make sense? So, you know, I don't need you to worry too much about quantal analysis. Quantal analysis is this, I think it's a cool technique where you're trying to figure out how much does one vesicle influence the EPSP, the excitatory postsynaptic potential of the postsynaptic cell. And usually, you know, something, I think this is a reasonable ballpark anyway, of about 0.1 millivolts per vesicle. All right. I messed up my numbering. Boop. Fixed. Nope, not fixed. Anyway, I think we're on question 3, though, aren't we? The thing says 4, but I think that's wrong. Okay, answer these questions based on the discussion on pages 133 to 136 of dendritic cable properties. I love this. So length constants. So the first one went to Chelsea. So what'd you get as far as what is a length constant? How does a length constant relate to the efficiency with which signals are transmitted down a dendrite? What's that last bit mean? Not sure? Okay. I was totally in agreement up to that point, but that was the one bit I wasn't quite sure what that meant. So the idea with the length constant, I like this description. It's basically how far do changes in membrane potential spread down, you said a cable, and that's technically true. But really it's we're talking about membranes at this point. So if you have a disturbance in one spot, how far does your disturbance spread? So I'm going to draw again. So there's this picture. I should probably just point at the picture, but I'm going to draw it. So, you know, the idea is that you're out on a dendrite. So this is supposed to be my dendrite. Here's my cell body over here. There's my axon coming across. That's pretty sloppy. But this whole thing's supposed to be the dendrite. And so we've got a synapse that's happening over here, where there's a presynaptic axon that's stimulating here. And the idea is that at this spot, if you measure at this spot, so here's millivolts over distance, so I'm using an x there for distance. At this one spot where you've got a depolarization, there's a pretty nice strong depolarization. The thing is, though, that this will decay, because if you're going along a dendrite, there's no voltage-gated sodium channels to open, so you can't regenerate your signal as you travel down the length of the dendrite. So it just kind of decays downward, sort of like this. The length constant is an indication of how rapidly you decay. And I think it's 63%, is that right? So somebody nod knowingly or correct me. Is it 63% of a decay? You're looking at me like you've never heard this before. It's 37%. Oh, shit. I think I got it backwards in my head. So you decay down. So if we sort of divide our thing in half. So if this is 37%, I shouldn't try to do this. I mean, basically what you're doing, it's until you get to 37% of the original. See, it is 63%. It's just, I think about it differently. Okay, all right, we're good. All right, so this would be 37% of the original signal. So you're basically looking at how far away from this original spot on the dendrite can we get before we're all the way down to 37% of original signal. And that value, this length, that, this is this distance you can get, that value is your length constant. So the more slowly you decay, so if you've got a really nice membrane that doesn't decay quickly at all, that's my darker line here. I thought I had a different color. This one would have a length constant, that's more like over here, so it would be a longer length constant. Longer length constants mean you decay more slowly, and therefore signals travel further down the membrane. Make sense? So I don't need you to calculate them. Like there's an equation and stuff in there. But I do want you to understand the relationship and what it means when you've got a length constant. That the larger the length constant, the more the farther you get, basically. So the more efficiently your signal gets down. Because you're losing signal because the ions are leaking back across the membrane. So you're losing some of that original signal. The less signal you lose, the longer the thing spreads, the more rapidly the ions spread, the longer your length constant will be. So it's a really handy measure that allows us to understand something about these properties that we just got done talking about in the prior chapter, which are membrane resistance and internal resistance. So that's where we're going next. Let's see. So, Brooke, you got both of these. Sorry. How will increasing internal resistance affect the length constant? What did you get for this? Internal resistance or memory resistance? I think you got them reversed. So current flowing across membrane would be talking about memory resistance. So yeah, so as you I'm kind of doing this backwards. As you increase membrane resistance, that's going to decrease the amount of current that will leak back across the membrane. So that'll decrease your signal loss. And therefore you'll have a longer length constant. Does that make sense? So the whole thing about membrane resistance is that you're trying to make this bigger. See all these people, everybody's like squinting because it's too small. And the light. Yeah, thank you. Sorry, I'm not being very helpful. Okay, so yeah, so the idea is your membrane resistance, this is basically telling you how leaky is your membrane. More resistant membranes are less leaky. So if you have a more resistant, higher membrane resistance, you'll have less signal loss due to ions leaking back across the membrane. And therefore your signal should travel further because it's more efficient. You're not losing signal as much. And therefore you get a nice long length constant. Does that make sense? So the other one is internal resistance. And I mixed you up. So like I kind of reversed you. Do you know, for internal resistance, did you have one? This is like, would decrease? I think that's right. So increased internal resistance Wait, what did you say? Did you say decreased? I'm thinking it out to myself. Yes. I got it backwards up here. That's why it wasn't working. Sorry. So internal resistance is the opposition to charge flow along a membrane. In this depiction, our goal is that charge does move across that membrane, across the inner surface of the membrane. So in order to get signal from the far end over here over to here, charge has to move. And so internal resistance is the opposition of that flow. So as we increase our internal resistance, this is going to result in slower charge flowing down the membrane. So it can't get down the membrane as quickly because of all that internal resistance. And as a result, you get decreased length constants. It just, your charge can't get there in any kind of appreciable rate. This is, this is lots of proteins running and bumping into the ions, and they're bumping into the cell membrane, and they can't find open space to move because the darn axons or the dendrites are too small and narrow. That's what's happening with internal resistance. Yeah. Uh-huh. It'd be less current. Yeah, I mixed that up. So increased internal resistance means you get less current flowing down the inside of the membrane, and therefore decreased length constants. Whereas if we increase membrane resistance, you'll decrease your signal loss, so your membrane is insulated, and therefore you get longer length constants. It sticks around. Which I think is what you were saying, Brooke, and I messed it up. So, sorry. All right. Let's do D then. So it says relate discussion. This to the discussion that we had in chapter 4 of 2 strategies used by animals to increase conduction velocity down axons. We had myelination, large axon diameter. So Tori, where are you? There you are. What'd you get for this? Agree, disagrees? So we had these two techniques we talked about. We could increase our membrane resistance, usually through myelination. That resulted in faster conduction down axons. And here we are getting back to this length constant idea that if you have more insulation, you'll have higher membrane resistance, and therefore you have a longer length constant. Again, that will result in more efficient transmission. You could also increase your axon diameter to decrease internal resistance, and therefore have a longer length constant. So I think that all fits very nicely. You feel okay on this? Length constants? Again, I'm not going to ask you to calculate them or play with that equation or anything, but I do want you to understand this, the relationship between membrane resistance, internal resistance, length constants, and the speed and efficiency with which materials can move down a dendrite or an axon. It's very important in dendrites because dendrites rely on that decay. I mean, they do not regenerate their own signal, and therefore the only way that they can get signals to go down their membrane of the dendrite is through this sort of decay process. Axons, you have a bit of decay, especially myelinated axons. You have a little of that decay that occurs in between each node of Rambier, between your Schwann cells or between your oligodendroglia. But again, the idea is that the better your lower your internal resistance, the better your membrane resistance, the more rapidly you'll get signals moving down. All right, let's keep on going. So this table tries to get a handle on the fact that we've got a gazillion different kinds of channels that we've been talking about over the course of the semester. And so the point of all of this is to sit down and try to parse out where are these things found and what are they doing? What is their function? So it asks you, I'm going to have to zoom out a little bit to get this all in here. So identify where you find each of these channels and what its function is. So Sam, you got the first two. Yep. Rephrase that slightly, but yeah. Are those okay? Anybody have anything different or additional? Yeah, so the idea is these things are all over the place. And I would agree, like they do play a role in these processes. They're not really, they're kind of open all the time though. So like the potassium leak channels, I'm just going to add a thing. So the potassium leak channels are always open. They're found all over your cell, all over your neurons. And because they're always open, they're always resulting in potassium ions leaking out of cells because potassium's at high concentration inside. Certainly it can leak back in, but the net movement is out of the cells. And therefore, they're always causing that sort of negative depression on the memory potential. There are sodium leak channels, and they are also open all the time. And they're always allowing sodium ions to leak into cells and make them inside of the membrane more positive. The thing is that in pretty much every neuron and every other cell, there's lots and lots of potassium leak channels and a very small number of sodium leak channels. And so at rest, when all these other channels that we'll get to are all closed, there are many, many more potassium leak channels open than sodium leak channels. So our memory potential is usually pretty negative at rest because the potassium channels just overwhelm the effect of sodium. Those all look great. All right, let's do voltage-gated ones, and these are maybe the fun ones. So Josh, you got this. Josh is not here. Okay. I don't know. Anyone want to go? Should I just talk, or I'll play Josh. So So the voltage-gated sodium channels, these guys are found on axons, and that's pretty much the only place they're found. You might find one or two occasionally in some other spot, but for the most part, voltage-gated sodium channels are only found on axons. These open at threshold to cause the depolarization upswing of the action potential. I should put the rising phase. That's probably, I think that's what we called it. To cause the rising phase of an action potential. The voltage-gated potassium channels are also found all over axons, and really just all over the axons. Those are the excitable membranes. These guys open slower than the voltage-gated sodium channels. But then they cause the falling phase of the action potential. So, you know, the big downward arc, as the voltage-gated sodium channels all inactivate and the voltage-gated potassium channels all open, you get a nice falling phase. I should also add the undershoot. So the reason we are more negative than usual in the undershoot is we have this larger than usual potassium current present during that time due to all these open voltage-gated potassium channels, and therefore we're closer to potassium's equilibrium potential. I would say it's both. Yeah, because so the repolarization, repolarization versus hyperpolarization. So the falling phase is the repolarization. We could get a falling phase by just inactivating the voltage-gated sodium channels, but it wouldn't be as quick. It would be a slower repolarization. So they're definitely speeding up the repolarization, and then they're exclusively responsible for that hyperpolarization undershoot step. That's a good question. Last ones are the voltage-gated calcium channels. These guys are different, right? So these ones are only found at axon terminals. And that's the only place, at least, that we've talked about them being. They open in response to action potentials. I can't type and talk at the same time. They open in response to action potentials, and the calcium ions that come in interact with proteins, the snare proteins, and cause vesicles to be released, to fuse with the membrane and then release their neurotransmitter into the synapse. So that's their job. I'm sure there's some electrical impact, but the electrical impact is not the important thing here in terms of causing membrane potential change here. The important thing that these guys do is they let calcium in, and it serves as this intracellular signal that interacts with snare proteins and says, hey, time to release your vesicles, and that's what they do. Okay? That's for neurons anyway. I mean, if you take a physiology class, you talk about heart cells, and heart cells have calcium channels that are involved in action potentials and do all kinds of weird things. But for neurons, the calcium channels, that's all they're doing for us. All right, the last two are the ligand-gated cation and ligand-gated chloride channels. What would they be doing, or where would they be found? So these ones went to, where are we? I don't even know where we're on. Oh, yeah, here. These went to Amanda. Where are you? Yeah. Okay. Yeah, I like that. Fix all my typos. Everybody agree, disagree? Is that okay? Yeah, so like there's all kinds of different channels that we'll encounter. These ones are maybe the two immediate ones that we talked about that are both receptors and channels. So the first one, this would be like an acetylcholine receptor. As we'll see, it's actually a nicotinic acetylcholine receptor that causes inward flux of these cations, and therefore, if it's an inward flow of positively charged ions, it'll cause an EPSP. The ligand-gliated chloride channels, these might be like a GABA receptor, and they open chloride channels once GABA binds and causes EPSPs. Sweet. And this would be happening, you know, like you said, postsynaptic membrane. So this could be on a dendrite, which would then have to, you know, the signal would have to decay down the dendrite to finally get to the axon hillock, or it could be right on the cell body, in which case it doesn't have as far to go before it gets to the axon hillock. All right, we got two more, and we have 3 minutes, so we're probably not going to get them all done. We can get this one done, though. So it says, gives you all kinds of stuff to read, but it says, using information from the pages listed in figure 6-7, describe a neuropharmacological experiment that you could do to determine whether receptors for acetylcholine on the second neuron are the same as receptors to the target organs. What did you do? This one went to Laura. Oops. I know I got a little bit off of your actual wording, but I know everybody's copying stuff down, but I'm going to flip over. So here's this picture, right? So the cool thing about these receptors, so these are both receptors, nicotinic receptors, muscarinic receptors, are both receptors for acetylcholine. But different cells will have different kinds of receptors. So some cells, some of these neurons may have a nicotinic receptor. Skeletal muscle has nicotinic receptors. Muscarinic receptors are found on other cells. Like you mentioned, the heart is one example of where you find muscarinic receptors. And while both of them are stimulated by the same neurotransmitter, acetylcholine, so you know if you expose acetylcholine to both these cell types, you can't really tell the difference in terms of their reactivity. The cool thing is we have found that there are agonists, meaning stimulatory molecules, that are specific to one versus the other. If you give nicotine, it's the same nicotine as found in tobacco, to a cell, and it gets stimulated by that nicotine, that means it's binding to a nicotinic receptor, which is the classic acetylcholine receptor, ligand-gated channel, opens a channel, cations stream through, all that jazz. On the other hand, if you take the heart, for example, and expose it to nicotine, nothing happens. Like it doesn't get stimulated because nicotine has no effect on this kind of receptor, even though acetylcholine will bind to it, just like acetylcholine binds here. This receptor, though, can get stimulated by this stuff called muscarine, which is some other compound derived from a mushroom. And then you can even do more, like you could do antagonist experiments too. So there are blocker drugs, atropine, for example, that blocks muscarinic receptors. Every once in a while, you'll be watching an ER show, and they'll say, "Give me 5 cc's of atropine." You know, that's why, because they're trying to block these muscarinic receptors that are found and present on the heart. That blocks the effect of acetylcholine. Acetylcholine slows down the heart rate, so they're trying to get the heart rate to speed up by blocking the effect of acetylcholine. But atropine does nothing to these guys. Instead, you can use this stuff curare, which I think is like from poison dart frogs or something. Is that right? Which is an antagonist for nicotinic receptors. So you can use this pharmacological approach to figure out what kind of receptor you have present in these different kinds of cells. All right, we are out of time. I went a minute over, so I will see you guys